Hey guys, welcome back to my YouTube channel Med So we have finished with the endomembrane system. Now let's look at other organelles. Firstly, we talk about mitochondria, the powerhouse of the cell. It is sausage shaped, or we can say it is cylindrical. So it is generally not easily visible under the microscope because they are very small. They are only 0.2 to 1 micrometer in diameter and 1 to 4 micrometer in length. Number of mitochondria per cell is variable. It depends on the physiological activity of the cell, so it is variable, not constant. By physiological activity, we mean the energy requirement of each cell. It varies. So the number of mitochondria is also varying per cell. Mitochondria is double membrane bound structure. So it has an inner membrane which forms the cristae towards the matrix. The cristae is nothing but infoldings. So where else have you heard of this term infoldings? Yes, we heard this term in mesosome while discussing about mesosomes. So they are infolding. Cristae is the inner membrane. They are infoldings towards the matrix. So the main function of cristae is to increase the surface area just as mesosome. So here there are two membranes, outer and inner, and these two membranes have their own specific enzymes associated with their function. So the matrix here is the cytoplasm of the mitochondria. It possesses single circular DNA molecule, and a few RNA molecules. So there is only one DNA molecule and a few RNA molecules. Note the difference. And ribosome of 70S type, smaller than the cytoplasmic ribosome, and other components required for protein synthesis. So remember, the DNA is single, double-stranded, circular DNA, important. And the ribosome is 70S type, which is composed of 50S and 30S subunits and few RNA molecules. And they have their own machinery to synthesize their own proteins. And this mitochondria is capable of autoreplication. It divides by fission, so it can replicate on its own. So it is the organelle capable of its own division. So important. So I told you mitochondria is known as the powerhouse of the cell, which means that mitochondria is the site of aerobic respiration and thereby producing ATP, energy currency of the cell. And it's important to remember that this aerobic respiration is associated with the inner membrane of the mitochondria, not the outer membrane. Why? Because it has specific enzymes. The inner membrane has specific enzymes required for ATP synthesis, aerobic respiration. Now we move on to plastids. Plastids is an organelle found in all plant cells and euglenoids. Euglena is commonly known as the plant animal because it has got plant-like features and animal-like features. It is phototropic and heterotrophic. So we say that the nutrition in euglena is mixotrophic. So more about euglena we will learn in the chapter of biological classification, which will come later on. So now we'll talk about plastids. So plastids, I told you, it's found in plants and it is easily observed under the microscope. Why? Because they are large. So this is the similarity we find with mitochondria, because mitochondria was small and it was not easily observed under the microscope. So based on the pigments in the plastids can be classified into three: chloroplast, chromoplast, leucoplast. Just by the name chloro, we find that the green pigment chlorophyll is associated with it, and with the name chromo, we know that they have colored pigments like yellow, orange, red associated, and leuco means colorless. Next, chloroplast, as I told you, it has chlorophyll and also other carotenoid pigments, important. So when they say carotenoid pigments, it does not mean it is only associated with chromoplast. Carotenoid pigments are also present in chloroplasts, and these pigments are responsible for trapping light energy for photosynthesis. So both chlorophyll and carotenoid pigments are present in chloroplast, remember. Next, in chromoplast, there is fat soluble carotenoid pigments again, like carotene and xanthophyll, which is responsible for producing this yellow, orange and red color. And leucoplast, there is no pigment associated with the leucoplast, and the main function of leucoplast is storage of nutrients. So where is chloroplast present? It is found in the mesophyll cells of green plants. Mesophyll cells are present in the leaves. Now chloroplast develops from chloroplast. Remember, this is generally observed during fruit ripening. For example, we take the example of tomato, ripening of tomato. Tomato is green, and when it ripens it becomes red. So when it is green, there is more chloroplast in it, and when it is red after it has ripened, all the chloroplast has converted into chromoplast by the extensive synthesis of carotenoid. The chlorophyll and the thylakoids get degenerated. So this is how the conversion happens. Talking about leucoplast, I told you main function is storage of nutrients. So accordingly, it gets modified into three types. First one is the amyloplast, which is to store carbohydrates or starch, for example in potato. Next is eleoplast, LIO. It stores oil or fat, example in mustard seeds. Next is aleuroplast, which stores proteins, example maize. We have seen in NCERT there is a structure of maize seed, monocot, embryo, that you can find aileron layer, which is triploid. It is a protein rich layer. From that we find that this name aileron, aleuroplast, is nothing but to store proteins. Okay, let's talk about chloroplast more in detail. It can be lens shaped, oval, spherical, discoid, ribbon shaped. So different shapes are present, and it is big, I told you, so it's easily observable in the microscope. So it has a length of 5 to 10 micrometer and a width of 2 to 4 micrometer. So these shapes you can remember by the super trick, which is LORDS, LORDS. Yes, L for lens, O for oval, R for ribbon, D for disc, and S for spherical. So with this you can remember the shapes of chloroplast. Okay, and chloroplast, the number, it is 1 per cell in Chlamydomonas, which is a green algae, and 20 to 40 per cell in Mesophyll. So it varies in different bodies. So just like mitochondria, chloroplast is also double membraned, and it has an inner chloroplast membrane which is less permeable. So the outer membrane is more permeable, inner membrane is always less permeable. Same thing even in mitochondria. So what are the terms associated with chloroplast? First thing is stroma. Stroma is nothing but the mitochondria, the matrix or the cytoplasm of the plastid, the chloroplast, is known as stroma. So let me draw for you. It has two membrane, I told you, outer and inner, and this matrix, the cytoplasm of chloroplast is known as stroma, and matrix, we told, matrix was for mitochondria. Matrix was the cytoplasm of mitochondria, and stroma is the cytoplasm of chloroplast. Next we have thylakoids. Okay, these are the thylakoids, and all these thylakoids together, the pile of coin, this structure you see, this is known as the grana or intergranal thylakoids. Both mean the same. Next we have connecting structures known as stromal lamellae. stroma lamellae, which connect the thylakoids of different grana. Next is lumen. So lumen is the space, can you see which I am drawing now? The space enclosed by the membranes of the thylakoid. Okay. So now where are the chlorophyll pigments present? They are present in the thylakoids and not in the stroma. So what are the similarities between chloroplast and mitochondria? Recollect and tell. One, they are double membraned. Next, they were double stranded. Single circular DNA was present in the matrix in the cytoplasm of the organelle. Next, they had 70S ribosome and they were capable of synthesizing their own proteins. And they also possessed a few RNA molecules. They are capable of auto-replication, that is, they divide by binary fission. So, they are known as semi-autonomous organelles. So, because of this feature of dividing by binary fission of the property of auto-replication, they are also known as bacterial symbionts, because we know that bacteria is the one that reproduces by binary fission. So, similarly, because chloroplasts and mitochondria are also able to divide by binary fission, they are known as bacterial symbionts, and they are also known as autonomous organelles, semi-autonomous organelles, because they can synthesize their own protein. They have their own double-stranded single circular DNA. So, and they possess RNA molecules and ribosomes. They are known as semi-autonomous organelles. Important. So, the stroma of chloroplast contain enzymes required for carbohydrates and protein synthesis, which means photosynthesis. Okay. It also contains small double-stranded circular DNA and ribosome, that is of 70S. Now, what are the dissimilarities of mitochondria and chloroplast? Chloroplast was big, mitochondria is small, and photosynthesis occurs in chloroplast and aerobic respiration occurs in mitochondria. So, these two were the points of differences. So, photosynthesis or photooxidation of light occurs in chloroplast, and chemooxidation occurs in mitochondria. That was all. Mitochondria was colorless and chloroplast is green in color, obviously. So these were the differences. Alright. Now we have something known as cytoskeletal elements, which is unique for eukaryotes. These are elaborate network of filamentous proteinaceous structures which consists of three components: microtubules, microfilaments, and intermediate fibers. These are present in the cytoplasm and it's collectively known as cytoskeleton. So what is the function and why are they present? It is for mechanical support, motility, maintenance, and shape of the cell. They provide shape of the cell. So double S double M: shape, support, motility, and maintenance. So if you want to know the differences between the three, microtubules, microfilaments, intermediate fibers, in terms of occurrence, structure, the proteins present, and the functions it carries out, you can quickly have a look at this table which I have provided, which is very easy to remember and has all the important points. You can have a look of this, but this is not present in NCERT, so you can skip this also. Now is the time to talk about cilia and flagella. They are hair-like outgrowths of the cell membrane in eukaryotes, whereas in prokaryotes, flagella is the extension of cell wall. Remember, that was the difference I told you in my earlier video. And cilia and flagella both are covered by plasma membrane, the cell membrane, and they emerge from centriole-like structure called the basal bodies, which this point also I have already mentioned while talking about the differences of flagella in eukaryotes and prokaryotes. So what is the difference between cilia and flagella? Cilia is something small. They work like oars, but flagella is longer. Cilia cause movement either of the entire cell or the surrounding fluid. We observe in our respiratory tract itself and in our fallopian tube there are cilia present which help in the movement of, for example, in respiratory tract it helps in the movement of mucus, right? So cilia is present there. Flagella on the other hand is responsible for the movement of cell, the entire cell from one place to the another. Cilia and flagella, this is an important topic, so pay attention to it. So this difference between prokaryotic and eukaryotic flagella I have already mentioned. About the flagellin protein also I have already mentioned. So go through the previous video carefully. So this is the topic, that is cilia, flagella, centrioles. This is a place where many students feel the difficulty. Otherwise this chapter is very simple. So pay attention. So first I am drawing the structure of cilia and flagella. So with this we can understand and learn the terms of it. So first of all there is a central doublet. Okay, there is a central doublet. So there is only one single thing which is in the center, and it is surround this is surrounded by a hub, and here you see there are peripheral doublets which are radially present throughout. So there are 9 periphery doublets, and these peripheral doublets are connected to one another by structures known as the linkers. So the 9 peripheral doublets are interconnected by linkers, and these linkers are known as the AB linkers. So you see the peripheral doublets, there are two segments, A and B. So the linkers emerge from A to B. So that's why they are known as AB to B. So the peripheral doublets are nothing but made of microtubules. So from the central hub, okay, there is radial spoke emerging, connecting the central hub to the A microtubule. Okay, so this structure which is connecting the central hub to the A microtubule is known as the radial spoke. Okay, so this is the central sheath, and this entire thing together is known as axoneme. So this has the arrangement of 9 plus 2. Why 9 plus 2? 9 doublets, 9 peripheral doublets which are radially arranged, and there is a singlet in the center, one, okay, which has 2 microtubules. So it is 9 plus 2. Understood? 9 peripheral doublets radially arranged, and the central, one pair of central microtubule which is present. So we represent it as 9 plus 2. So now let's read the text and understand the meanings and terms. So the core is known as the axoneme, I told you, which has several microtubules running parallel to the longitudinal axis. So what is several microtubules? So what is the total number of microtubules? I told you 9 plus 2 arrangement. So 9 peripheral doublets, 9 into 2, okay, plus the 2 central pair, okay, the one central pair of microtubules. So 9 into 2 plus 2 is 20. So this axoneme has 9 doublets radially arranged peripheral microtubules and a pair of centrally located microtubules. Yes, clear? And this arrangement of axonemal microtubules is referred to as the 9 plus 2 array or 9 plus 2 arrangement. Clear? The central microtubules is connected by bridges. Yes, there are 2 central microtubules. Both of them are connected by bridges. This is known as the bridge. Okay, understood? And is also enclosed by a central sheath. Yes, we saw that there is a central sheath around that one pair of microtubule in the center. Yes, which is connected to one of the tubules of each peripheral. What is that connected to? One of the tubules of each peripheral doublets by a radial spoke. Yes, what was that microtubule? That was the A microtubule. So the central sheath is connected to the A, that is one of the tubules, that is the A tubule of the each peripheral doublets by a radial spoke. So how many radial spokes are there? There are 9 radial spokes. So I hope now you have understood. And the next text says 9 peripheral tubules doublets are interconnected by linkers. So there are two terms here, three terms here: bridges, linkers, and radial spoke. So bridge is something connecting the microtubules together, okay, and linkers is the one connecting the peripheral doublets together, and radial spoke is connecting the central sheath to the A microtubule. Understood? So can we move on to centrosome and centrioles topic? Yes. So centrosome is an organelle which has two cylindrical structures known as centriole, okay, and it is surrounded by amorphous materials, and these two centrioles lie perpendicular to each other. So I will draw it so that you understand. So this is the centriole, this is the kinoplasm or the centrosphere, the matrix, okay, and this pericentriolar material, the amorphous pericentriolar material or maculae, okay, this is amorphous which is covering the entire thing. Understood? Now are you able to understand? So this two centrioles are together known as diplosome, which are perpendicularly arranged to each other. So centrosome is nothing but diplosome plus kinoplasm, which is together covered or surrounded by the maculae or pericentriole material. Understood? Now are you able to visualize? So can you say this has a cartwheel like organization? So this information is about centrosome. Now we'll talk about these two structures, centrioles, which lie perpendicular to each other. Now let's talk about them in detail. So each centriole, if you see under a microscope, they will have a cartwheel like organization, and they are made up of 9 evenly spaced peripheral fibrils of tubulin protein. So they have 9 tubulin protein which are peripheral fibrils, okay. Each peripheral fibril is a triplet. So in cilia and flagella there were peripheral doublets. Now here there are peripheral triplets, and similarly adjacent triplets are connected by linkers, like how adjacent doublets were connected by linkers in cilia and flagella structure. And the central part of the centriole, which is a proteinaceous structure, was known as the hub here. There the central core, it was known as the core, and the entire structure was called the axoneme. Understood? And here again there are radial spokes made up of protein, which is connecting the central hub to the peripheral doublet. So likewise I will draw the structure so that you'll understand. So this structure is the hub, and there are 9 radially arranged peripheral triplets. Okay, these peripheral triplets, each of them is made up of a fibril. Okay, each triplet fibril is made up of a protein known as tubulin protein, and there is no central sheath like the cilia and flagella. There is only the hub over here, and this hub is connected to each of the peripheral triplet by radial spokes, and this you see is the linkers. So I told you there were A and B microtubule, but here it is A, B, C because they are triplets, isn't it? So the 9 peripheral triplets are made up of tubulin protein, which is absent in the center. Center, do you see, is there any central microtubule present? No, there is only a hub present. There is no microtubule here. So what is the arrangement, you say? It is 9 plus 0 arrangement. There is nothing in the center. Understood? 9 peripheral triplets made up of tubulin protein, but the triplet fibrils are absent in the center. There is only a single hub present. So the linkers are nothing but CA linkers. Okay, there it was AB linkers. So try to differentiate and learn. It will be easy for you. You won't get confused. And how many radial spokes are present here? There are 9 radial spokes and there is one central hub present. So I told you centrioles form the basal body of cilia and flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cell. So centriole give rise to basal body and this basal body gives rise to cilia and flagella. Understood? But centriole and cilia and flagella, they are totally different structures. Centriole has 9 plus 0 arrangement. Cilia and flagella has 9 plus 2 arrangement. So you should understand. Okay. Centriole gives rise to basal body and basal body gives rise to cilia and flagella. So very important to understand in this place. Okay, please pause and try to understand what I am trying to tell you. Next, centriole gives rise to spindle fibers and spindle fibers gives rise to spindle apparatus. So these are the two important functions of centriole. Centriole is absent in plants. It's present only in animals. Important for cell division. How? Because centriole gives rise to spindle fibers and spindle fibers gives rise to spindle apparatus. So this flowchart makes everything simple, makes all the lines given in your NCERT simple. So this is a super trick. You can remember this in a flowchart so that you'll never forget. Okay, so enough about centriole, cilia, flagella and centrosome. Now let's talk about the next organelle, which is nucleus, the control room of the cell. So nucleus, I already mentioned earlier that it was first described by Robert Brown. So you tell me who is Robert Hooke. This also I have mentioned in my previous lecture. Okay, nucleus of the interphase. So interphase is a phase of cell division, okay, cell cycle where the cell is not dividing. Such a phase is known as interphase. So about cell division, cell cycle, you will learn in the very next chapter of botany, which is very interesting. So stay tuned. So in such an interphase nucleus, what are the components? Let's have a look one by one. First one is the chromatin. Chroma means color, okay, just by the name. It was named by Fleming and it got stained by basic dyes. So since it got stained by colored dyes, okay, chroma, that's why the name chromatin. So it is stained by basic dyes. Remember, not acidic dyes, they will confuse you. And it is highly extended and elaborate loose indistinct network of nucleoprotein fibers. So chromatin is nothing but it is a loose, something like thread like, very loose, and they are not present as distinct structures, they are indistinct, and it is a network of nucleoprotein fibers. Chromatin contains DNA and some basic proteins called histone proteins, and there are also some non-histone proteins and RNA present. So what does chromatin contain of? Chromatin is basically a nucleoprotein fiber. So chromatin has DNA, it has RNA, it has histone proteins, which is basic, and non-histone proteins. So important. Okay, histone, non-histone, DNA, RNA. Okay, all this comprise the chromatin. So a single human being, we know they have 2.2 meters of DNA in each cell. Okay, that is distributed among its 46 or 23 pairs of chromosomes. Understood? We already know this basic common logical information that we have 23 pairs of or 46 chromosomes in each cell. Next structure of the interphase nucleus is the nuclear matrix or nucleoplasm. Next is the nucleolus. There can be one or more in each nucleus and it is spherical structures, remember. So more about nucleolus, let's see. So the content of nucleolus is continuous with the rest of the nucleoplasm. Why? Because nucleolus is not bound by any special membrane. Okay, and it is a site of active ribosomal RNA synthesis or RNA synthesis. So there are different types of RNA, mRNA, tRNA and rRNA. Okay, this you will learn in the next grade, grade 12 you will learn. So nucleolus is not a membrane bound structure, it is the site of rRNA synthesis. Remember, So that's why nucleolus is known as ribosome factory. So by this term ribosome factory, we do not mean that nucleolus is a site of ribosome synthesis. No, nucleolus is a it's a site of synthesis of rRNA, ribosomal RNA. That is why it is known as ribosome factory. So be careful. It does not mean ribosome synthesis happens. It means ribosomal RNA synthesis happens. So I told you one nucleoli or many nucleoli can be present in each nucleus, and nucleoli can be larger and more numerous in cells which are actively carrying out protein synthesis. So rRNA gives rise to proteins and gives rise to structure known as ribosomes. They get associated, and ribosomes I told you is known as the protein factory. So don't get confused between what is known as the protein factory and what is known as the ribosomal factory. Nucleolus is more abundantly present in cells which are actively taking part in protein synthesis. That is also an important point to remember. Next component of the interphase nucleus is nuclear envelope. Nuclear envelope consists of two membranes which are parallel to each other, and these parallel membranes has a space between them which is of 10 to 50 nanometer. 10 to 50 nanometers. So the units is also important. This space is known as the perinuclear space. This space is important because it is forming a barrier between the materials which is present inside and outside this nucleus. Inside the nucleus and outside, which is the cytoplasm. This is also a double membraned organelle, remember. And the outer membrane is continuous with the endoplasmic reticulum. I told you endoplasmic reticulum is an organelle which is present just in close association with the nucleus, and it has ribosomes on it. And in many places nuclear envelope is interrupted by very small minute pores. These pores are known as nuclear pores. So how are these nuclear pores formed? They are formed by the fusion of two membranes. So this space is not present. The two nuclear membranes fuse at certain points, and this results in a pore-like structure known as nuclear pore. Nuclear pores are also very important because they help in transport, movement of RNA and proteins in both directions in the between the nucleus and the cytoplasm. This both direction is very important, an important term. It's just not in one direction. So about this perinuclear space, it is forming a barrier, and about nuclear pores, it enables movement of RNA and proteins in both directions. These 2 points are extremely important. So nucleus, there is only one nucleus per cell in most of the cells normally, but there are organisms in which there are more than one nucleus present. For example, in paramecium, it is a ciliated unicellular protozoan which has two nuclei. One is the macronucleus and the other is the micronucleus, and these two nucleus has particular functions to perform. Next is fungi, which has ribosome and penicillium. Both of these have two nuclei, more than one nuclei present, a multinucleate condition. And talking about humans itself, animals, skeletal muscles. Yes, we know they are the voluntary pair of voluntary muscles. They also have multinucleate condition. This we say it is structural syncytium. Whereas in fungi like rhizopus and penicillium, we call it cenocytic condition. So both these terms are referring to multinucleate condition arises when karyokinesis is not followed by cytokinesis, the cenocytic condition. So remember, there are two terms which I used here. So there are other cells in mature state, they lack nuclei. So what is this condition known as? enucleate condition, because in mature state they are lacking nucleus but not the earlier stage. So that is RBC. Yes, we have already looked at this example. And sieve tube cells of vascular plant, they have enucleate condition and not anucleate. Anucleate means nucleus itself was absent throughout its life. Okay, so we talked about chromatin, which is present in nucleus, which is present in the interphase nucleus, but it gets condensed and results into distinct structures known as chromosomes during other stages of cell division, like other than interphase. So this chromosome, okay, every chromosome is visible only in dividing cells, okay, cells which are dividing. When cell is not dividing, it is known as interphase cell. In such interphase cell, chromosomes are not present, chromatin is present. So each chromosome has a primary constriction or the centromere, and on both sides of centromere there are disc-shaped structures known as the kinetochore. Centromere holds two chromatids of a chromosome. So let me draw so that you will understand. So what are the terms we saw? Primary constriction or the centromere, which is this structure, and this centromere holds two chromatids of a chromosome. So one chromosome has two chromatids, and on either sides there are disc-shaped structures called the kinetochores that help the attachment of spindle fibers during cell division. So there are four types of chromosomes depending upon the position of where the centromere is present. Metacentric, meta means middle, the centric means the center part of the chromosome. What is the center part of the chromosome? It is the centromere. Yes. So meta, middle, centromere. So the name tells you that centromere is present in the middle. So the two arms, the two chromatids are equal. Next comes the submetacentric. Sub means little, slightly away from the middle. Okay, resulting into one shorter and one longer arm. Next comes the acrocentric. Acrocentric means closer to the end, something closer to this end. Centric means your centromere. So the centromere is located closer to the end, forming one extremely short arm and one extremely long arm. The short arm is known as P or the petite arm, and the long arm is known as the Q or the Q arm. Okay, let's not look into the Latin names of what Q and P means. Okay, P means short. That much if you know, it's enough. So where are the short and long arms present? It's present in submetacentric as well as acrocentric chromosomes. Next, talking about telocentric. Telo means towards the end. So the centromere is present at one of the end. So there is a terminal centromere. Understood? So simple, the four types of chromosomes. In few chromosomes, there are certain non-staining regions called the secondary constrictions present at constant location. So please look at the terms that I am using. Chromo means color, I told you. So the chromosome structure, they get easily colored by basic dyes, I told you earlier. These chromosomes have non-staining regions called the secondary constrictions at a specific constant locations. So this location does not differ from one chromosome to another. In all the chromosome, whichever has these secondary constriction, they are all present at the same constant location. This gives the appearance of a small fragment called the satellite. So satellite is a fragment and the region is called secondary constriction. So remember, so this chromosome here is the secondary constriction. This constriction you see here is the secondary constriction. NOR, non-staining constriction. NOR means nucleolus organizing region. Okay. This region gives the appearance of a small fragment which is projecting here. This you see is the satellite. So what is this? This is the central part, primary constriction, centromere. The end of the chromosome is known as the telomere. Telo means, I told you, terminal, end. So the end of the chromatid is known as the telomere. So one chromosome has how many telomeres? Two telomeres. So this is all about chromosomes, chromatid and nucleus. Let's talk about the other microbodies, the other organelles, small bodies present in the cell, and this completes the chapter cell, the unit of life. So micro means, I told you, they are minute vesicles. They are membrane bound. That is, they are bound by plasma membrane, and these contain various enzymes. And microbodies can be present both in plants and animals. There are basically three types of microbodies. One is the peroxisomes involved in peroxide synthesis. Next is the spherosomes. Tell me what is spherosomes in the comment box. Is it plant lysosomes or plant Golgi bodies? I have discussed about dictyosomes and spherosomes in my previous video, so have a look. And there are structures known as glyoxysomes. So this is all about microsomes, so microbodies. So you just have to learn the information given in NCERT. So I'm not going into too much details. So let's stop with this. So this forms the end of the chapter cell, the unit of life. So let's not end it so easily. I'm just giving you a simple homework. I want you to prepare a list of membraneless, single membrane bound and double membrane bound organelles. Okay, so this is your homework and please comment it down. So that completes the chapter. Thank you so much. Subscribe if this video helped in your learning process.